DNA and Replication by Alana Kyer. A strand of DNA is made up of molecules called nucleotides. A nucleotide has a nitrogenous base, a sugar group, and a phosphate group. The four types of bases a nucleotide can have are adenine, thymine, guanine, and cytosine, but in RNA, uracil replaces thymine. The nucleotides are attached to strands of DNA opposite of each other. These also attach to create the sides or rungs of the double helix. Adenine bonds with thymine and cytosine bonds with guanine, or adenine bonds with uracil in RNA. The sugar and phosphate groups bond to make the sides. The DNA molecules are very long, so they are coiled up and tightly packed into chromosomes. Each chromosome has 2 strands of DNA inside of it. Enzymes. There are 3 enzymes that are involved in replication. The 3 are helicase, DNA polymerase, and ligase. The helicase unzips the strand of DNA into 2. The DNA polymerase bonds the 2 nucleotides, and the ligase fixes breaks in the backbone of the strand. Replication forks. A replication fork is where the strand of DNA breaks apart for replication. In prokaryotic cells, there is one point of origin that splits into 2 forks, and they move in the same direction. In eukaryotic cells, there can be multiple forks that are moving unidirectionally. Having more points of origins and more forks can help make the replication process proceed faster. Direction of DNA polymerase. DNA polymerase can work toward the replication fork on only one strand, or the leading strand, while on the other strand, the lagging strand, it must proceed away from the replication fork. The lagging strand breaks into Okazaki fragments that are later combined to form a full strand. When in a cell's life. DNA replication occurs during mitosis in the S, or synthesis stage of interphase. Cells spend most of their lives in interphase before mitosis occurs. Semi-conservative model. During DNA replication, an original parent strand separates, and each daughter strand is used as a template for the synthesis of a new second-generation daughter strand. This results in the formation of 2 identical copies of the original double-stranded molecule. This is what we call semi-conservative replication. Primers. A primer is a short strand of DNA or RNA that serves as a starting point for DNA synthesis. It is required for DNA replication because the enzyme that catalyzes this process, DNA polymerase, can only add new nucleotides to an existing strand of DNA. Okazaki fragments. Okazaki fragments are short, newly synthesized DNA fragments that are formed on the lagging strand during RNA replication. They are opposite to the lagging strand, together forming short double-stranded DNA sections that are eventually connected to complete the full strand. Repair and proofreading. Replication has some sort of built-in error detector. Sometimes during this process, the wrong nucleotide will be added to the strand of DNA. If this happens, DNA polymerase will detect the error and replace the wrong nucleotide with the correct nucleotide. With this function, billions of errors are detected and corrected. Thank you for watching.